Okay, so welcome everyone to this new session of the BioClock Academy. It's the 11th edition today. My name is Laura Pape and I'm a PhD student at the BioClock Consortium, and together with Oana Georgiana Roos-Oswald, I'm going to present or moderate this session today, host it. So the BioClock Consortium, really shortly, as you probably all know, is a very large Dutch research consortium focusing on research surrounding chronobiology and the biological clock, as well as in human research, also animal research in society, in different surroundings. And this BioClock Academy is a product of the BioClock Consortium. We do this every month, so every third Wednesday of the month. We invite senior researchers to explain to us basic concepts and equip you with proper knowledge and theoretical knowledge about chronobiology related topics. And today we will have a 40-minute talk followed by a 15-minute opportunity to ask questions. And so roughly 1 hour, we will close at 5 again. And during the talk, you can also write your questions in the chat if you'd like, or after the talk, you can also just speak up for yourself. So I give the mic to Georgie.
Hello, everyone. From myself, my name is Georgiana. I am a postdoctoral researcher in the BioClock Consortium. And today I want to introduce you to our guest speaker, which is Professor Marcel Visser. He works at the NIO, the Netherlands Institute of Ecology. His research mainly focuses on understanding how climate change disrupts natural systems by using long-term studies on wild species such as birds, for example. He's involved in many exciting projects as far as we could research, for example, collecting and analyzing longitudinal data of bird populations, but also trying to build entire ecosystems to enable research studies in testing various climate scenarios and forecast possible outcomes. And with this, I don't want to take too much out. I guess Professor Visser will tell us more in his talk. I want to give the virtual floor to him. Welcome, Marcel, and thanks for being here.
Okay, thank you. Thank you very much for inviting me. I think it's an excellent idea to have this kind of academic series for all the different disciplines that are associated with clocks. So I'll share my screen. Okay, well, so today I will talk about seasonal timing. And as was requested by the organization, I'll try to give a bit of a broad overview over seasonal timing. And I want to start by talking about a paper that we published really quite a long time ago now, in 2010, where we talk about phenology, seasonal timing, and circadian rhythms. And all these different terms describe the same thing, but they're being used in a slightly different way by very different research disciplines. So I just want to go to the different perspectives of this of the same phenomena and try to explain how they relate to each other, and I will present a framework in which we can study those. So if we start with phenology, phenology is defined as the study of periodic life cycle events and how these are influenced by seasonal and interannual variation in climate. So if you talk about phenological phenomena, you can think about things like bud burst of trees, egg-laying days of birds, returning birds to their breeding areas, but also the end of diapoles for insects. Basically all the things that you see happening in springtime. Although phenology can also refer to the things that happen at this time of the year in autumn. For instance, birds returning back to the wintering grounds, or the leaves of trees that turn red. These are also events that happen every year, roughly at the same time, but not exactly at the same time. And that's where these seasonal and interannual variations come from. So for those of you from the Netherlands, you might be aware of the Natuurkalender, which is an initiative run from the Wageningen University. And there people can send their observations, their phenological observations to the Natuurkalender. And then they will give you a kind of an overview of how other people have reported that same phenological event this year or in previous years. And Arnold van Vliet has been running this now for probably 2 decades or so. And it's a nice way of people to contribute to science in a kind of citizen science way. And this is a not very typical example because this is probably the longest running phenological time series in the world. This comes from the cherry blossom trees in Japan. It's an important cultural phenomena in Japan, and therefore it's been recorded for a long, long time, over 1,200 years now. And what you see here on the x-axis are the years, on the y-axis the date of the cherry blossom. And what you see is that there is a lot of variation. But up to the mid 1800s, it was kind of a stable, always around the 15th of April. The advancement we see after that, we'll talk about a little bit more later on in the talk. But for now, I just want to point out that there's a lot of variation from year to year. And that is what phenology is about. It's about when does this happen for the whole population at a particular time, associated with, for instance, temperature changes or rainfall changes. So this is the phenomena to be studied, but also evolutionary ecology study the same phenomena, but they don't call it phenology. They usually call it seasonal timing. And they refer to these as these phenological events, seasonal timing, and the emphasis is very much on the variation among individuals within years. So one individual will have one of the cherry trees will have a flowering date which is earlier than another cherry tree, which might be in the same location in the same year. And they also put a lot of emphasis on this between year variation. But rather than just describing the between year variation as it happens in phenology, they also want to understand that between year variation and ultimately that the year to year variation has to be associated with changes in the environment. So in some years it's good to be early, and in other years it's good to be late, and therefore the evolutionary ecology will usually talk about seasonal timing in terms of trade-offs, constraints, and fitness. So this is a very simple figure that describes how evolutionary biologists look at seasonal timing. So in the blue curve here, you see the fitness of the consumers. So birds that lay early have a low fitness, birds that lay later have a higher fitness, and birds that lay even later have a low fitness again. And this now is a distribution of what the birds are actually doing, or the animals are actually doing. And what you see is the individuals that are early have quite a high fitness. Individuals that are a bit later have a lower fitness. But if you're really late, you will have a really low fitness. So in this case, what you would see if you would go around your study site, that there seems to be selection for earlier events. The earlier you are, the higher your fitness is. And this is quite often found. So this is the way that the evolutionary ecology will look at seasonal timing. But seasonal timing is also studied from more like a physiological level. And there the questions are very often about the how question, the causal mechanisms. The physiologists want to know how do the environmental cues like temperature and photoperiod, how does it lead to change in morphology, physiology, and behavior during the year? And traditionally, physiology has really emphasized the effect of photoperiod. They're very detailed studied how the increase in photoperiod in springtime leads to changes within an organism that then sets in motion, for instance, reproduction. Also quite interesting about physiology is very often only males are used. So, for instance, then the parameter that is being analyzed is tested with size variation, for instance. So the onset of the reproduction in males. Now, this is an example of just one of the many examples of how seasonality can be can be causally arranged within for instance, in this case, a bird. So you have environmental cues, and the clock plays a role in the brain and hypothalamus, for instance, that then lead to hormone secretion, LH, FSH, that has an effect on the gonadal development, and then in the end it leads to sexual behaviors like egg laying or reproduction or courtship. And of course, there are also negative feedback loops that keep the whole thing shut the whole thing down after reproduction has been over. So this is something that physiologists would study in great detail. And one of the interesting things about this system is that temperature plays a big role very often in phenology. So in early warmer years, things are usually earlier, and cold years, things are usually later. But it's quite unclear how that works. So this is a paper we wrote quite a while ago as well, in a very nice section of the PLOS Biology journal, which is called Unsolved Mysteries. So one of the unsolved mysteries was then, how does temperature influence the seasonal timing in endotherms? And are some ideas about it that we also lay out in the paper, but I think at the moment we still are not completely sure how that works. So then another group of people that work on seasonal timing are the geneticists, the molecular geneticists. And they try to do two things. They try to understand genetic variation among individuals. So some individuals will have a different genetic background than others, and it might be that those also have a different timing, seasonal timing, and that then can lead to selection. But there's also a way, the molecular genetics is also a way to understand the causal mechanism underlying seasonal timing or underlying phenology. So if you know which genes are associated with early laying and with late laying, it will also give you an idea. It will help the physiologist to understand how the mechanism is shaped. And this is a typical example, for instance, for great dates, late dates, where you have, in this case, about 500,000 SNPs, so markers on the genome, and you try to find an association with the variation in those markers and the trait you're interested in this case, late date. And you plot them per chromosome here. The different colors are different chromosomes. And you look at that association with the genetic variation and phenotypic variation. And because you do so many tests, you have to correct for the number of tests you do. And that's the line you can see here. That's the line above, if points are above that line, there's a significant association, correcting for the number of tests that you're doing. And what you see is that none of the points actually reach that significant threshold. So this graph is typically called a Manhattan plot because you're supposed to see big skyrise and high peaks for the genes that are associated with your trait. And normally for many of these timing traits, you find more like what we call, there's no high rise. It's all kind of Uppsala plot with only low buildings. In this case, you don't find an association with certain genes and the phenotype you're interested in. And you can use the molecular genetics also in a very different way. So if you look to the left, this is also by graded late dates where we made a selection lines for birds that were late layers and selection line for birds that were early layers. And we did a selection not so much on the phenotype, but we did a selection on the genome directly. So for that, you need a training population. So you had a training population of about 2,500 female gradients with a known lay date, and roughly 500,000 SNPs. And then what you can do, you can select every year, every generation, you can genotype the offspring and select the offspring, those were the early genome and those were the late genome, and that way make selection lines. I'm not going to say more about it than this because there were 3 PhD students that wrote their thesis on this, so it's impossible to summarize this in 3 minutes. But if people are interested, they can either read the papers or we can have a chat about it after the talk. Well, and then finally, then the chronobiologists, because the chronobiologists, of course, also study annual and daily fluctuations in physiology and behavior. And they're called circannual rhythms because they roughly happen every year, circannual. And traditionally, chronobiologists focus on the internal timing programs, and that enables organisms to cope with and also anticipate mainly uh, uh, changes in the environment. So that can be either changes at the level of the day-night. Many of you might be working on that, but you can also anticipate, of course, changes that happen on a more annual timescale. Well, this is, I think, the classical example of that work on circannual rhythms. This is by Abel Gwinner. He kept a bird, a stone jet, in 10 years under the exactly same conditions: 12.8 hours light, 11.2 hours dark, constant temperature. And if you keep birds under those conditions, they will still cycle. So the red here in year one, the red you see, this is gonadal size. What happens in birds is that in wintertime they have very small gonads, and then when springtime arrives, their gonads start growing, and then they have a maximum size gonad. And then in after the breeding season, so say roughly July, the gonads become smaller again, and then they're small again throughout the winter. And what you see for this bird that was kept on these conditions for 12 years is that you see that cycling every year. You see the gonads growing. And this is the mold of the feather mold that happens at the time of shrinking of the gonads. But what you also see is that these peaks are not at the same place in every year. If there was a perfect annual cycle, you would see that this increase and decrease would happen always at the same time of year. But that's not happening. The bird is free running because it doesn't have any cues about where it is in the annual cycle, the outside annual cycle. So what you see, it starts free running, and this bird has a tau, a free running rhythm, shorter than 12 months. And that's why you see that this period of gonadal increase and regression starts shifting over time forward over the year. So this is a very similar graph as probably many of you have seen for day-night rhythms, where you have free-running birds or free-running mammals in a setup where they have no information about the day-night situation, and they free run depending on their free-running rhythm. So this shows very clearly that there is an internal clock that is also worked at the annual level. We don't know exactly where that clock is. There is definitely work done on that. But what we do know, of course, is that this clock can be modulated by, for instance, photoperiod. And here you see the example of a starling. In the top panel, the light cycle is exactly 12 months. So you see again here, the gonads are small in wintertime, they grow and then they regress again, and then they grow and they regress again. Over 2 years, there are 2 cycles. But if you now make these light cycles shorter, 8 months, 6 months, 4 months, 3 months, even all the way down to 1.5 months, you also see that the cycling of the gonadal size starts being compressed by that. So clearly, photoperiod is a very important cue for birds to know where they are in the outside world calendar date, and they also respond to that. But they also respond to temperature. And if we look at, we have studied that quite extensively in climate-controlled aviaries. We have 36 climate-controlled aviaries here at the NEO, where you can keep a pair of gratids and you can give them a specific temperature pattern and look at their seasonal timing. So here you have the lay date on the y-axis and the 2 treatments, cold treatment and a warm treatment. Our treatments are not necessarily a particular temperature, but we mimic specific years. So we have a very cold year, was 2013, very warm year, 2014, and we mimic those temperatures exactly. So every hour, the climate controlled aviary gets a different set value, and therefore you really mimic the temperatures as they were recorded outside in those 2 years on an hourly basis. And that's because we don't really know where the information is stored in the temperature pattern. We don't know where it's the minimum temperature, the maximum temperature, the variation in temperature. So by mimicking those temperatures exactly as they were recorded, we hope to retain all the information that is in that temperature pattern. Each line is an individual. We can ignore the colors here. These are the early and late lines I talked about a little bit before with the selection lines, but it's not so relevant here now. And half the birds got first a cold spring and then a warm spring, and a year later we flipped the treatments. And what you see is that on average, these birds lay earlier in the warm year than in the cold year. So that means that there's a really causal effect of temperature where birds lay earlier on the warm conditions. Now, I want to make now a kind of a shift to something which is called phenotypic plasticity. I'm not quite sure how many people are well aware of the phenotypic plasticity. So I'll give a bit of an introduction to it. So what we just saw was we had lay dates on y-axis and in that case temperature on the x-axis, some environmental variable. And these are then the phenotypes that are expressed by a single individual under these different conditions. So this line is called the reaction norm. It's the relationship between the trait, in this case lay date, and the environmental variable, which could be temperature. And that reaction norm has two characteristics. It has a slope, the sensitivity to temperature, and it has an elevation. There could potentially also be a line that runs below that. And that tells you something about lay date in the average environment. Now, phenotypic plasticity is defined as the property of a genotype to produce different phenotypes in response to different environmental conditions. So it's the same genotype, and in this particular case are the same individuals, but you bring them in different conditions, different temperature conditions, and then they express a different phenotype, in our case, a lay date. But it's not necessarily only about seasonal timing. It could also be morphology, behavior, other life history traits that responded. Phenotypic plasticity is very, very common. And sometimes it's only once in a lifetime that something is developed, and sometimes it happens multiple times during a lifetime. And for instance, lay date in great teeth is an example of this multiple lifetime situation where the same individual will live for a number of years and the condition will differ from year to year and they will respond to that. Now we can try to we can think about phenotypic plasticity as being adaptive. And this is what I try to explain in this slide. So suppose you have three different environments, red, green, and blue environment. And these are these fitness curves. So this is a bird that lays early in this environment, for instance, low fitness, a bird that lays it a bit later, higher fitness, bird that even a bit later, and even more later, low fitness again. So the optimal thing to do in this red environment is to have your trait value here to be. For the green environment, the optimal value is here, and for the blue environment, the optimal value is here. And now what I do on the right-hand graph, I flip this. So sometimes it's difficult for people to get this, so I'll try to explain this carefully. In the red environment, it's best to be early. So your optimal value is a low value. In the blue environment, it's good to be late. So your optimal value is a late value. So now environment is on the x-axis and the optimal thing is on the y-axis. So in the red environment, it's good to be early, and in the blue environment, it's good to be late. And this is now the reaction norm. So it looks very similar, but something very different. The reaction norm is what the animals actually do. And this is what the animals should be doing. If they follow this line, they will always hit the optimum in every environment. And this is what the animals actually do in reality. Very often the reaction norm is less steep than the optimal line. And that's because there's very often uncertainty in exactly what environment you are. So you have these kind of optimal relationship and the reaction norm. Now we can try to put this all in a kind of a conceptual framework, and that's what I've done in this slide. So I'll walk you through this and try to explain this in detail. So in the middle, we have our phenotype, in this case, laydate. And how does laydate come about? That's on the left-hand side. Of course, there's a genetic component, but there is also an environment that is temperature, maybe photoperiod, maybe internal clock. And these things together, the genes and the environment, are in a response mechanism, and that leads them to laydate. And that response mechanism, that is of course where the phenotypic plasticity is. Yeah? So a genotype expressed a different phenotype in different environments. That was the definition of a phenotypic plasticity, and that's exactly depicted on this side of the diagram. So in a warm year, birds will lay early, but there might still be birds with genes that are even earlier or genes that are late. Now, then on the right-hand graph, we see that laydate then is being selected for. That is translated into fitness, translated into how many offspring do you have, what's the condition of this offspring. And that relation between the laydate and fitness, that depends on the environment at the time of selection. In the particular case of gradients and breeding, it's when there's a lot of caterpillars around because the birds feed their offspring with caterpillars. Now, it's important to realize that the environment at the time of decision making and the environment at the time of selection are not necessarily the same environment. So birds need to produce their eggs. They lay one egg every day. They need to incubate these eggs for 12 days. Then the chicks are born. So there is roughly a month between the time that they make that decision, that laydate decision, and that they have the chicks in the nest. And therefore, the environment has changed quite dramatically in that month. So in this environment, they probably look at things like photoperiod, their internal clock, temperature. But here, the time that there is a lot of caterpillars is also affected by temperature, but not necessarily the same temperature. So the one thing you need to remember from this diagram is this arrow. There has to be some logic. What are you using here? You're using here the things that predict this environment. If temperature does not predict this, you should not be using temperature. If photoperiod does not predict this, you should not be using photoperiod. And therefore, this example of photoperiod, clock, and temperature is true for the gradients, but it's not generally true. It's not true for any species. So if you look at a zebrafinch, a bird that lives in Australia in the desert of Australia, when does it breed? It breeds when there are enough seeds of the grass. That's it, because it needs seeds of the grass for its offspring to feed. So it doesn't care about temperature. It doesn't care about photoperiod. It only cares about rain. Because if there's rain, there will be grass. And if there's grass, there will be grass seeds, and then it can feed its offspring. So a zebrafinch will not use temperature or photoperiod as a cue. It will use rainfall as a cue. And the crossbills are even a more exceptional species because the cones of these trees are used by the crossbills to feed their offspring, the seeds of those. And the crossbills are a kind of a wandering species. They when they find a place where there are cones, they start breeding. So they use the cones as their cue because it's tightly associated with the food that they're going to give their offspring. So the cue use, there are no general rules for cue use. You need to know what is predicting the food that you're going to use later on for your offspring, for instance. Okay, so all of this is what I described is a general theory, but now we're going to apply this to a changing world, a world that is rapidly changing. For instance, land use change. For instance, climate change, for instance, urbanization, and of course, more relevant maybe for this audience, the light at night, the light pollution. So all these things are changing quite rapidly. And how does it now link in with this whole seasonal timing? So I'll first give you an example more from my own work, which is about increasing temperatures. And here, this graph, you've all seen this graph in one form or another. This is the temperature on the planet relative to the 1951-1980 mean. And this is year, and what you see, of course, is since the 80s, temperature have been going up, and it seems to go up even faster nowadays. So what does it do to seasonal timing? Well, this is to come back to that phenology being phenotypic plastic. I explained it just now. So this here, every point here is a year for my long-term study. So you see that if it's cold in springtime, the birds are late when they lay their eggs, so end of April. And if it's very warm in springtime, you see the birds lay their eggs much earlier already, maybe around the 15th of April. And this relationship is because individuals respond to temperature. As I've shown already with the climate Avery example, they respond to temperature directly. And you see that the different lights are different individuals. Different individuals might differ in their timing, but they all respond to temperature. If you now look what happens in the forest, you see that the great tits, they change their lay date over time. They become earlier and earlier with about one day per 3 years. But if you look at their food, as I said, what's important for the great tits is the caterpillars because they feed that to their offspring. You see that advances its phenology as well, but twice as fast as that for the great tits, about 2/3 of a day per year. And this pattern that we see for the great tits and their food falls in very well with a much more general overview of many species of organisms, primary producers, plants, primary consumers like the caterpillars, secondary consumers, the predators of those primary consumers. And this is the change in phenology in days per decade. And what you see is that for these primary producers and the primary consumers, they shift roughly 4 days every 10 years. But these secondary consumers, like the great tits, they only shift half of that, only 2 days for 10 years. So what you see is that those shifts that we observe due to increasing temperatures are not the same. The shifts are different. The shifts are mainly in the primary consumers and the primary producers, and less so in their secondary consumers. So phenology is temperature dependent. Climate change leads to higher temperatures. Phenology advances, but not all phenology is advancing at the same rate. And therefore you get what we call phenological mismatches. The birds are now too late. So climate change leads to what we call ecological relationship problems. Everything is changing, but not everything is changing at the same rate. So these are a couple of examples from other things. This is roe deer or caribou. These animals, their offspring are born in springtime. At that time, the grass has to be very young and contain a lot of proteins because the mothers need that protein to produce milk for their offspring. But what you see with the grass starts growing earlier and earlier nowadays. But the day that the birth date of the caribou and the roe deer is not changing. So the roe deer and the caribou, of course, the time that they conceive is very early, very late in autumn, and there are no real cues available for them to know whether it's going to be an early or late spring. So their birth dates not change very much, and the grass changes quite a lot in their phenology. This is a slightly more complicated example. but it does show how complicated it can be sometimes. So this is a bee, and the male bees come out of hibernation in springtime earlier than the females. So at that time, males are flying around looking for females, but the females are not there yet. And this orchid makes use of that. It has flowers that mimics a female bee. And the males try to copulate with that female with those flowers, and thereby they pollinate the flowers. But nowadays, because of increasing temperatures, the bees are getting earlier and earlier, and the female bees are getting earlier as well. So now, by the time the orchids are flowering, the female bees are out already. And of course, the male bees are slightly more interested in female bees, in real female bees, than in those flowers. And this is an example of barnacle geese. The barnacle geese, they overwinter in the Netherlands, but they breed high up in the Arctic. And of course, at the time that they depart from the Netherlands, they do not know anything about phenology in their breeding grounds. It's just too far away. There are no cues available in the Netherlands that are informative about how the world looks like a couple of thousand kilometers north where their breeding grounds are. And therefore, what you see happening now is that they might leave a little bit earlier, but not much earlier. But the condition of breeding grounds move forward much more strongly. And therefore, by the time they now arrive at the breeding grounds, they still need to produce the eggs and incubate the eggs. And by the time their offspring are born, the grass that is available for the offspring that used to be matched with the time they were offspring, now the offspring is later than when the grass is out. And this is another example, but this is not so much about animals and their food, but this is more about males and females doing different things. So the female Arctic ground squirrels enter hibernation earlier, but the males do not change their hibernation earlier. So this was a paper in Science of this year. The graph they give is a little bit strange because they give two lines and you think, oh, there's a males and females, but it's not the case. It's just two locations. Both lines are for females, and they don't give the line for the males. They just give the p-value for the change in the males, and that is not significant. So the females are changing, the males are not changing. And of course, that also leads to relationships problems, but then between pairs. And the last example I want to show is even more extreme because this is about changes in different life cycle stages within the same individual. So, for instance, male arriving by flycatcher, it is very strongly dependent on photoperiod and about condition in the wintering grounds in Africa. But their egg laying date of the female depends very strongly on temperature in the breeding grounds. So you see the female egg laying shifts forward, male arriving is not shifting forward. So that leads then to less time here. But you can also see that now mold moves forwards as well, and that leads into an overlap with the chicks. So this even moves more forward than the laying date and the chick hatch date. So you see that within the life cycle of an individual, different components shift at different rates because of climate change. Okay, so why? Why does climate change lead to differential shifts in phenology? Why is not everything shifting at the same rate? And for that, we go back to the graph, to the schematic I showed you earlier. And you see that temperature plays a role here at the side of the response mechanism, but temperature also plays a role here because it affects to some extent when there are going to be caterpillars. And now the problem of climate change is that climate change is not affecting these temperatures in the same way. So temperatures early spring are increasing, but not as much as temperatures in late spring that are affecting the caterpillars. So here you see the temperatures relevant for the birds that runs from mid-March to 20th of April, while the temperatures are relevant for the caterpillars run from also early March to the 14th of May, mid-May. And you see these temperatures here are increasing more than temperatures there. So climate change leads to a problem because it's affecting different parts of the year in a different way. And that's why this is changing not as rapidly as the slope here. Simply because the temperatures that are relevant for the birds in making the decision are increasing to a lesser extent than temperatures that are relevant for the caterpillars. Okay, then for the final few minutes, we go back to the light at night situation. So can we now also think about this in terms of artificial light at night? So for that, we have to go to the experimental site of the Light on Nature project that is run by Camille Spoelstra. We have set up lamps in the forest. I'm sure some of you have seen these figures before. So they are either dark control, there's green light, there's red light, and there is white light. And there is nothing else in that forest except those lamp posts. So we are sure that any effect we find are due to the light. And we have nest boxes hanging around these transects, and there we can look at lay dates of ratings. And if we do that, we find that the lay dates of birds in the dark control is later than for the green and the white light. The red light is not significantly different from the dark control. So it's clear that there is an effect of artificial light at night on when birds are laying, so on their seasonal timing. We have done that in 6 years, and those years are here on the x-axis. And here what you see is that those differences that we observe in panel A are due to two years. The 2 years that were very late. So these were cold years where everything was late. And then we see that the lay date of the white and the green in particular are affected by the light. They are earlier than the dark control. Well, in these other years, the years that things were relatively early, the warmer years, there is no difference. So we find an effect of light at night, but only in conditions where everybody is late. And then probably light plays a more important role, an overriding role in their time. So going back to this diagram, now this is not about climate change, but about light. And of course, the key question that you all have now, of course, is, okay, I've shown you now that light affects to the environment at the time of decision-making lay dates, but is light also affecting when there's a lot of food in the forest? And is that now also earlier, because we see light lead to an earlier lay date, and perhaps light will also lead to an earlier food peak. So these are a couple of years where we looked at caterpillar abundance and when there's a lot of caterpillars in the forest. And here you see the 4 colors again. And what you see that indeed in the white and the green light, there is much more food than in the red and in the dark control. But it doesn't seem, although it's not so easy to see because there's not such a clear peak in the dark control, it does not seem that there is also an advancement of that peak date. Here it's maybe a little bit, but it doesn't seem to be that there is really a strong advancement of the food peak. So the effect that light has on the decision of the bird to be earlier might not be adaptive in the sense that also when there is light at night, the food is going to be earlier. Okay, so that was it. So this paper, I guess, might be interesting to read. It will tell you a little bit more in detail what I've tried to bring across today. That although phenology, seasonal timing, and circadian rhythms come from different disciplines, they talk basically about the same thing, and we can provide a kind of a framework in which we can try to combine all these things within a single framework. And then finally, this is something that I wrote for the United Nations Environmental Program. It also talks really about that seasonal timing, the rhythms of nature, and how it's being disrupted by climate change. And it's much more also very accessible for non-specialists. Okay, that was it. Thank you.
Thank you very much, Marcel, for this interesting talk.